of the transfer RNA. So the best correlation to number of physicochemical parameters at the second base was UCGA, and you can describe it at the unit interval using both characteristics. You have subintervals, and you can specify 0, 0, 0, 1, 1, 0. Depending on, we are going to observe row to column representation, so first row, second column, or second column, first row, and of course the last was the third one. We obtained different parameters. Empirically, it is known that second column, first base, so column row, correlates well with the physicochemical parameters, while row column calculation of distances by coin tossing positioned on the unit interval correlates well with coevolutional parameters. But this correlation is somewhat weaker than the one in the first one. So this is the representation of the combination 1, 2. However, despite of the weak correlation to physicochemical parameters, somewhat weaker, it is still very predictable and can be used for very precise calculation of the secondary protein structure, because if we use a standard error control code like linear block triple check code, we actually have 4 bits information which contains all data and correction for the third base. We published that in 2003, and at that time we were not fully aware where the last 2 check bits follow, and they basically lead into a relational model. Of course, when you calculate a distance or position of the unit interval, you have an algorithm for 6 digits, and depending on the dimension, as Branko showed yesterday, you can use that classic dyadic one. You can use exponent of 3 to be, you have a Bannekanter set, a ternary one. So one can manipulate with algorithm and the dimension calculation. 6 is usually enough for binary and Cantor, so for exponents 2 and 3, it covers well. So the structure itself, Y20 amino acids and 64 codons out of that 61 for the coding, and 3 for stops, and Y45 transfer RNAs in average. In this lecture, we will of course focus on the relational data model and look at those reasons within its frame, and show, of course, that it's a general pattern for all codes. The relational model is the most commonly used model of data storage. It uses highly optimized tables and the largest language structure consistent with the first order predicate logic. It was done by Edgar Codd in 1970. In 1983, Codd obtained Turing Award for this, and it was commercially applied for the relational database systems, for example, or like Oracle. and the language used is SQL. So in the context of terminology, relational model proposes the distributed storage of data into a collection of tables, and the basic elements are rows and columns. Rows are called also tuples, records, and columns and attributes. Now, let us go back to the standard genetic code table. First, we have, as we have seen, to decode it, which is basically means that we have to switch G and A table, because if we start in succession, U is 1, 0, 0, C 0, 1, G is, and not A, is 1, 0, and A is 1, 1. We published a long time ago, in 2001 and 2000, that this basically means that we are dealing with a gray code. And Rosemary Swanson also noticed that in her paper. It was one of the first papers on gray code, as far as I know, the first one which sensibly shows the whole system. This is not the problem. We also see how Wes's amino acid polarity requirement correlates well when we do this concerning columns. Wes also noticed the column situation, and indeed, when we put it like this, it's a proper arrangement. Now we can go further. The first step is a normalization of the table, which means that we have introduced, according to obtain relational first normalized form, it's called the first normalized form, key columns, that's the first and the third base, and non-key columns, which is the second base and contains attributes or data in the context of amino acids, or stop codons can be also treated in that way. So then the next step is to remove subroads, but first I'll show those three tables, how we have key columns, non-key columns. This is the first base, this is the third base, and non-key columns are data attributes, in this context amino acids. Now when the first normalized form is done, we are going to eliminate subrows to establish a primary key. Primary key is important, and to remove the transitive dependency. Transitive dependency means that between the third base is eliminated as a component between the first and the second base. Basically, this is done in a relational model by seeking for so-called joint columns. It is a column that is present in all four tables, and that column is, of course, column C, as we see. So, when we construct all four tables, we remove transitive dependency, define joint column, we obtain the second normalized form. We can track the system from top or top down, or for all triplets to simple structures. So, in the biological sense, we have 4 16-field 1-atomic tables, in which the third base defines the table, because the third base is each of the table. So we have first, second base table for U, first, second base table for C, first, second base table for A and for G. Second base defines columns, first base defines rows, and in SQL sense, it's third base is from, select, where those are instructions for the system. Now the result of the model is that we can manage the data taking care of the logic relationship, truth function, Boolean functions, and Cayley table, which in this model we applied is a Klein four groups Cayley table. Concerning amino acid origin, we have two phases, amino acid of the two phases, phase 1 and phase 2. The phase 2 is biosynthetically sourced, and the phase 1 are prebiotically sourced. And when we observe, I put all four tables together in one, we see that we have some boxes with duplicated amino acids, because in the first two tables you see you have F, and in AG you have L, and so on. But the constant one, the constant structure containing 8 amino acids, so 8 is in addition to joint column, this box CU, GU, CG, and GG, and this structure resembling Klein cross is actually a Romer's diagram. Now it looks something different, somewhat different. You have a cross structure, which you don't have it in the normal genetical table, simply because G and A columns and rows are changed. So all is now put into its position. I will skip this. And okay, Kleinian groups are a useful tool for manipulating with 4 operators. We have non-identity elements, element and identity elements, of course. 3 non-identity elements produced 2 of them produced third one. It basically shows XOR operation in the context of truth function or technical devices and so on. And we can represent it in a digital numerical form as partly permutation representation of subgroups. I will skip some of the, maybe to show how it looks codon-anticodon relationship. It's also complementary. And of course, this red field, these 8 red ones, which are numerals, it's invariant part of the code. In those parts, in that part, you have only one amino acid which is specified in all 4 tables. So 4 tables are optimal representation, and concerning transfer RNAs, UNC tables have the same, basically identical, and 2 separate transfer RNAs are sets are made of A and G in the context. So we have UC is identical, and G and A. So we can count them all, minus 3 stop codons, it's 45. And this corresponds to known RNA transfer RNase number, which is estimated to be 45. And it's on the level of 16 doublet codes. So, one of the assumptions that is evolved in small steps, mosaical structure. There are some questions: is it frozen, accidental, or what is going on with Luca? And now we showed it, we investigated it for all 33 possible codes. And for U and C, nothing happens. Except one anomalous, we can say, exceptional code in yeast mitochondria, in specific yeast mitochondria, but it's the only exception for leucine. The other part remained almost intact, practically intact. For the third, base G, something more happens. Some new amino acids are introduced. And in the A table, no new amino acids are introduced. We see that stop codons are the whole U row. This is also an in-join column for one specific code. There is S is becoming stop codon, and this is also an exception. So we have two exceptions in the whole set of all genetic codes, and there are exceptions within some of those codes. So the exceptions are also the yeast mitochondrial codes. So in some yeasts, we have two exceptions for leucine and serine in joint column. All other do not show this change of pattern, variant pattern. Of course, around there are all we have distributed. We see that this column G, it's actually a column that Julia wrote a lot about. It's a column of the evolutional code. So joint column and column of the evolution code are practically intact. So they're accept. Now, what's really happening here? We see that the amino acid seems to be introduced in one table after another. Of course, the problem is also with the fact that we have redundant amino acids. Also, one very interesting feature of the genetic codes, which is seen here, that serine, leucine, and arginine are redundant. However, there is a difference between third and U and C third base, which are identical, serine only, and G and A with leucine and arginine. So this makes it happen in the context that as the transfer RNAs are different, in the same manner, the redundancy of amino acids is differently, the information on the amino acid redundancy is differently contained for serine in first two, and leucine and arginine in the second two. I'll skip to this. And now, what is going on in an evolutional context? So we have invariant part of the Klein cross. It's SP, we have its joint column part of G row, and basically Romer, as you see red. Now, with UC tables. To that invariant part, this part is introduced: F, C, Y, H, D, M, and I. So when this is filled, and we have practically no codes except this one anomaly here that makes any relevance or change or anything. Now in G table, the rest of amino acid is introduced, resting M, W, Q, E, and K. And it's finished with something going on around that core part, all mutations around, but at specific boxes. And finally, the third base, the second stop is introduced, and the system is finished, but in the third base, we don't have any changes. So we have invariant part, UC and G introduction of amino acids. Now, two-dimensional analysis of the system with those three groups enables us to make scores. And so we wanted to test it. Obtain two-dimensional matrix and calculated the distances to see phylogenetic trees. It was done in a standard way, and we used one essential protein for DNA repair. So REC-A is a phylogenetically conserved protein in all bacteria, archaea, and eukaryotes. So, and without it, there is no life. You cannot repair DNA. So DNA breaks are not repaired. A sample of 34 organisms from different domains is used. Analysis was done in a standard way with first muscle software, then Mr. Bass algorithm with EUG software, and here is the result. This rule, call it rule, really differentiates and clusters bacteria, eukarya, and archaea in the same manner as Wes's ribosomal RNA sequences were clustered when he made a hypothesis of the third kingdom. So basically we had three kingdoms here in a manner identical to Wes. I put the Wes's diagram to see this is his paper and this is REC-A. So this is about synthesis of protein, but this is about repair of proteins through evolution. All three kingdoms are fully in line with the relational model, with relational tables, and it's worked well. The other classic example we did is for hemoglobin data set. And we see here PAM 250 class standard matrix, empirically obtained. So it is used for phylogenetic analysis. And we see here the results of the relational model on the same. The patterns are identical. And invertebrates, hot and cold, mammals and others are well separated. We see here gorilla and at the top human and human sickle. It's one mutation which is typical and which changes the hemoglobin. That is the general idea of applying such things for any purpose. complex evolutional analysis. I will skip this. According to Barbieri, during the evolution, there is some optimization of system rules, and the ambiguity is eliminated and the system starts producing specific components. As we have seen, the relational model can observe, can be used for such differentiation. We have several phases how the amino acids from invariant part of the codes to ones depending on the third base, G column, G table or A table, are optimized for particular transitions in evolution which are based on biological specificity. The relational model is suitable to display the genetic code and its variants. Moreover, it enables the constructions of specific or slightly more different tables for some specific genetic codes. So it means that if we have a specific genetic codes, one of those, 33, or 32 actually, in addition to standard, we could analyze closely related organisms which use that code and more precisely observe the differences. So its applications are in code biology, in bioinformatics, and are fully in line with the concepts of Marcello Barbieri. That will be all. Thank you.
Thank you very much. We open discussion. We have.
Please accept my apologies for a possibly nasty question. My question is, couldn't you have carried out the same research and the same analysis out of the neo-Darwinian perspective, or possibly awareness that of the arbitrariness of codes helped you or pushed you to carry out this research?
I will address the neo-Darwinian concept in the context of Kimura's model in the next presentation. But basically, what I could say, we have seen here how the decomposition of interrelational model tracks how the code encoded amino acids from invariant ones which have fixed positions within in the context of relations in of the first and second base. On the other hand, there is another aspect. The Cayley tables of the operators itself, U, C, A, and G, and that's the part I skipped, those trees, because I wanted to not to show it twice, simply. And you will see that they also follow, a distribution of the Cayley table. So we are getting to the same evolutional conclusion from two different angles. One is a group combination within the Cayley table according to XOR algorithm. But XOR algorithm is a complementarity because you have strong bases and weak bases as two elements. weaks and zeros, strongs and ones. So the client table, it contains within itself as an algorithm the biological component of complementarity. Messenger RNA codon, and the information it's in its complement, which is contained of transfer RNA anticodon. So I'll show it. Yes?
Okay, thank you, Nicola, for a very interesting talk. I would like to ask, according to these, these 16 of the 16 codons analysis, can you say that at the beginning it was not coding with codons which had 3 nucleotides, but 2?
It's very speculated, difficult to speculate. Maybe it all went quickly. This seems to suggest that there were several mechanisms. Yes, first combining first and second, then the third, and first and embedding first the prebiotic amino acid, and then at some time point those who are synthesized. Well, I can say, and now we have some new result of it because Arenas and Lutz conducted this work, and those patterns can be extracted. So when we didn't have time to do it, to present it, because it's just finished, it works the same way. And it's fascinating that the conductances extract those patterns. Only isoleucine is in one row. Sorry, but I was just. That does not. This only changes slightly the length of the sliding block, so can be modified. This isoleucine is not a problem from the biological point of view because there are some metabolic pathways to bypass it. So it has a logical, biological explanation. And just one more, that column, G column, which Di Giulio find at Wadek, it was statistical proteins were done by a Japanese group, and they made random proteins out of it in an attempt to do statistical ones out of those 5 amino acids. And interestingly, they observed 2 things. They didn't fold accurately, they were statistical, but they had a characteristic that they were more soluble, significantly more soluble than the ones obtained for other rows. So this would, their work would actually imply that not only statistics itself from that the solubility is also very important for the proteins to enter into different metabolic pathways. We'll see that. Oh, sorry.
Thank you. Excuse me. Yeah. Okay. Thank you very much. Our time is moving very fast. And now he is the second. Yes, of course. Please.
It's in Italian,
Yeah,
so
yeah.
I'm
In
a bit
Italian, I know.
I'm
Presentation.
confused with Croatian
No, no.
as well. Everything
Yeah,
is in
yeah.
English.
No worry.
Okay. My second talk will be on the square opposition modeling in code biology, from biological codes to codes of culture. Why? Because we have some data that Anna did some time ago, and we were working together on them, me, Paško, and some time ago as well, and Albert also pushed the problem of semiotic squares in our focus. Because he was interested in a completely different problem of movements, but it turned out that this application turned out to be logical. Anyway, from the standpoint of the genetic code, the investigation of square opposition represents solution to some biological encoding problem, and it will be the first part. In the second part, we will present the square of opposition based analysis of a culture code, basically assessment of intrinsic and extrinsic religious motivation. We selected this religious motivation since religion is an assessment of human condition, culture, and it's linked to beliefs and background. We had an excellent and well-known HOG, intrinsic religious motivation scale, which is a standard psychological instrument that measures different ways of being religious, and it's applicable to a wide range of the groups. As of the square opposition, it is a simple diagram for the representation of basic categorical propositions from Aristotle Tractate on to Apuleius and Boethius in the 6th century. So for a long time, people, scholars, and philosophers were exploring the square of opposition. In traditional logic and the modern philosophy language, we usually name the basic proposition as A, E, O, and I, affirmative or negative propositions. And of course they are put in the diagram in the square opposition. In 19th century, George Boole introduced the version of square which resembles the modern one, and Gemma introduced the semiotics in the 60s. However, during the last decade, the considerable progress was done with respect to application of square opposition, not only in logic, but a true function application, but also in evaluation of some psychological scales, cognition, and basically practical application. So for the application of code biology of particular interest are the topics of truth function-based calculus with a square, client group manipulation with the square opposition and constructing algorithms, and analytical problems related to it. So I will just mention a few important highlights in the area. Vybranec-Skradowska shows in 2016 that truth functions can be efficiently used to define complex sentences or propositions within 16 binary truth functions, and it works well within the framework of classical propositional calculus. Regarding client for group and scale of opposition algorithm contracts, Sarasset and Dimai investigated logical geometries. So they investigated geometries and geometrical changes within square, transition to hexagon, or transition to more complex geometrical elements in order to make some application in artificial intelligence-based reasoning or description of the systems based on hexagons, for example, different geometric bodies, depending on the physical or mathematical objects that model the shapes. Some analytical problems were also addressed by Pfeiffer and Sanfilippo. They constructed the probabilistic versions of the square opposition and show that it is applicable in psychology, linguistics, and social science. They also showed how it can be used to define tripartitions of the set concerning coherent assessments on within the finite set of conditional events. So in a series of papers they were investigated service as well, boundaries of the intervals, and design of experiment, including some testing of visual ability, cognition, and so on. There is a large, in several papers, they also introduced truth functions and combined it, which is a logical thing since we have already seen that it works, with square of opposition and some extensions to hexagon of oppositions. So, as we saw in the first lecture, it is very important to encode the descriptive information of nucleotides and amino acids and proteins, and it can be used, we will show soon, for defining hydrophobic moment, intrinsic disorder, and atom depth. So some standard bioinformatic parameters can be defined using some truth functions and some error parameters like B factors, which measure the error within the measurement itself of the protein within PDB database from which you extract the structure. They also give B factors. So system modeling ranging from Miyazawa-Jerningham, for example, statistical potential, to Kimura 3 substitution type model, which is linked to evolution and non-Darwinian evolution molecular. A chemical nomenclature has been proposed for some time for nucleic acids. And however, this was not so practical for some single mutations and so on. And of course, amino acids and codon changes has to be tuned, as we have seen by the algorithm I mentioned before. So what we continued is to formalize the system and show how ambiguity codes specify those molecular descriptors and information flow itself. First, we use chemical nomenclature, defined it according to Klein-4 group and square opposition positions. Then the truth functions were specified for each element with operator symbols, logical operator notations, and logical operation itself. Now, when this all was put together and fitted according to the chemical description, truth functions were presented, and this is a classic Cayley tables, which represents in its essence a complementarity when presented as a Klein-4 group. It's an XOR system, based system. We can make it as with truth functions, binary form, numerical form, in the form of a Cayley table, or in the form of rows, columns or rows. Of course, the complementarity works for messenger RNA, transfer RNA complements. The truth function XOR becomes NOR. And all these things can be shown by using tables, either made of digital, numerical, operator positions, and works with Romer as well, diagram. So in the context of chemical nomenclature, the changes within the changes done by the client group define transition, transversion, transition, transversion, changes of all possible changes. And of course, those changes are expressed in the semiotic square. We published it earlier in the second issue called Biology, by affirmative and the negative conditions. Another point is that it can be also expressed by the probabilistic square of opposition with tripartition on the unit interval. So there is a lot of possibilities for the application of this algorithm and of putting different boxes of two-dimensional tables into correspondence to unit interval position. This is the picture of several of these functions. We have white scores and gray scores, true and false. So those patterns can be also used for calculation as zeros, ones. So these are all 16 patterns. This is for XOR. XOR shows the complementarity. The complementarity CG in UA discriminates. And in reality it works because thermodynamic patterns of bonds also follow this. We have an lower operators, high parts in the same manner as this. So these are 4 bonds, 4 bonds, 5 bonds, or 6 bonds. And it's confirmed by experimental data, and there is a very strong correlation, 0. It also works for mutation rings. And I will skip some of the data, and those elements can be also used to obtain bioinformatic parameters by using column data, column characteristics, column measurements. Chemical ambiguity code V, it's OR, truth function OR, and B factors. So from this we can have empirical RSA, Miyazawa-Jerningham classic statistical potential, atom depth, and all calculated very precisely and modeled very precisely with only a few parameters. Similarity, this problem was also done in the context of the Cayley group positions. Also with REC-A, we tested it in the same manner as I described earlier. In phylogenetic analysis was also done with muscle and UniPro. Interestingly, if we see no Kimura's paper on evolutionary distances, it is the same pattern as we have here. Now, here is almost turned from 90 degrees as semiotic square, probabilistic square for position. So it looks like two-dimensional analysis of Cayley tables can be done, matrix calculated from it, the values as 1, 3, 4, 7. We obtain also almost perfect clustering of bacteria, eukarya, and archaea, very nicely separated in the same manner as Wesse did it when he was explaining archaea. As well, the same situation is confirmed with Blossom and PAM standard matrices, so the system works. At least works well enough as Blossom and PAM, which are used for standard evolution analysis. Now, let's go to religiosity. We use hard linguistic religious motivation scale in the same manner to measure different ways of being religious. It's a famous and well-known scale, and Hodge was a very influential and well-known expert in religious studies in America. During his career, he wrote many books and was guided in the association of the Society for Scientific Study of Religion. When he did this scale, he didn't rely on Christian tradition only. he wanted to make a psychological item which is of a general value. And the instrument avoids potentially divisive issues on behavior, beliefs. So it actually measures the motivation behind religious activity. And probably because of this reason, the scale is widespread, popular, and different population and different researchers use it. So he produced actually an a unidimensional scale, relatively simple, with 10 questions, with the aim to provide clarity in the study of religious problems, issues, and factors of interest to social scientists. It was tested in many different populations. For example, it's valid for Muslims as well as for Chinese. It was also validated in Buddhism. So it's now considered as a general instrument. Recently, it was also validated in Portugal 2 years ago, 3 years ago. So it's basically a very popular scale. The questions are, there are 10 questions. Questions marked blue are extrinsic, intrinsic, and extrinsic are marked red. So 2, beliefs are less important, living a moral life, refuse to let religion into life, many more important things than religion. Those are typical extrinsic. The others are extrinsic. We will see that very important are, in my life, I experience the presence of divine. Nothing is as important as serving God, and religious beliefs lie behind my whole approach to life. Standard response system was used for this scale. I would evaluate this scale. Strongly disagree, moderately disagree, moderately agree, and strongly agree, which is perfect, of course, for the square of opposition-based analysis. The scale was validated also for Croatian population, a large number of examinees of similar age, sex, span. And interesting is that when we investigate the situation with cohonent neural networks, the extrinsic religiosity was extracted. So questions 2, 5, and 8. Beliefs are less important, refuse to let religion into everyday affairs, and many more important things than religion. We see that those questions and Q sticks from it. Principal components and clustering analysis also extracted 2 basic groups, extrinsic and intrinsic items. Now, scoring system was additionally applied based on serial opposition, and we done machine learning analysis of the responses to extract rules and variables relevant for accurate discrimination of examinees based on religious beliefs. We used VICA machine learning algorithms for data mining purposes, and and scoring system according to Klein foregroup. Within the system, we introduced numerical representation of the Klein foregroup, 1, 3, 5, and 7 for different elements. So the scores look like this. This is a probabilistic version. This one was a semiotic one, but basically they're the same. There's no difference. So this is the evaluations: strongly disagree, moderately, and strongly. I will be very soon finished because there are several classification algorithms we used. First one was one rule. It's a method who identifies the basic rule. Then C5-4 algorithm and part which is quite well often known and used, and the part decision list which adds additional validity to the system. In any way, we extracted 6 variables and 7 rules. In my life, I experienced the presence of divine, who was Q4, which was the first and ground algorithm. Then we applied C.4. It's a machine learning system, one of the best 10 algorithms based on entropy and information gain, which extracted Q4 and Q7. In my life, I experienced the presence of divine, and nothing is as important as serving God. So 94% of the people were correctly classified. It has a very simple tree. The best algorithm was part, and in the end, we can say that really extracted 2 good algorithms with either 7 or 4 rules, with only 3 intrinsic variables, Q4, 7, and 9, which enable a very precise discrimination of religious motivation of a person. This may have some impact on surveys because you need only 4 simple questions, Q4, Q7, Q9, and age. Of course, you can ask them sex as well, but it's not within the framework of the algorithm. So we can conclude that such an algorithms can be used based on square root deposition. Just 2 more points, very short one. Recently, Hopfield neural networks were thought to be core for the associative memory and general memory, have been made based on Kleinian foregroups, practically the same algorithm and this one. So this means that neural network constructions can be done at the same principles. We showed here that we can apply different concepts from ambiguity and codopoesis, Barbaciao, Barbieri, with a methodology similar to ones of Piper, who constructed probabilistic versions of the square opposition hexagon, and our methodology, which is similar and goes into a trout functions problem and defining. So we are at the moment with elements who can analyze both cultural codes and genetic codes with the same systems, even neural networks constructed in similar ways. Despite of a very much difference, as Barbieri remarked, between culture code and general code, this tells us that we have also some possible psychological and social design studies which could be based on the same elementary principles as the genetic code analysis. Thank you very much.
Unfortunately, we have 15 minutes in today, so we have obligation for the entire organization. So I ask for the next speaker. So here is your time.
Okay, thank you very much. So thank you to the organizers for inviting me to this exciting conference. It is very inspiring to hear talks from people from so many different fields. And I almost feel a little bit guilty of having prepared a talk for a rather narrow audience, or on a very relative scale. So I apologize in advance if some parts are technical and if I occasionally use a jargon of my field. So I'm going to talk about the gene regulatory code, and I'm going to do so very much by comparing it to the best known biological code, the genetic code. I do think it's probably the most important code in the biology of higher organisms, but I want to start with a somewhat provocative slide where I just want to summarize the state of decryption of the two codes. So on the left side we have the genetic code, we have the nucleotide sequence of the histone H4 protein sequence, and if you want to assess the status of the code, I would ask the question, can we predict the protein sequence from the gene sequence? And the answer is a clear yes, and we can do it with 100% accuracy and certainty. Now, if we do the same exercise with a gene regulatory sequence, I have chosen an ultra-conserved element from human, and here we probably should ask the question, can we predict the cell type specific effects on target gene transcription? And the answer is a clear no, we cannot say anything about it, and I insist anything. And so it doesn't mean that we don't know anything about the gene regulatory code. I would say our state of knowledge is a little bit like in the case of the genetic code, we are pretty sure that it's a triplet code, and we suspect tRNAs to be the adapters, but we have absolutely no meaning how codons relate to amino acids. I would say it like this, but also it's a warning that I'm talking sometimes as if we would know a lot about the code, but most things are highly hypothetical. So also I want to clarify what I mean by gene regulatory code. the activation of a gene, or the regulation of the activity of a gene, is the result of a very long cascade of molecular events. So there are events that happen upstream of transcription. So they include chromatin opening and closing, chromatin structure in many ways, DNA modifications, mainly methylations, and histone modifications. And then there is the key event transcription initiation. And if I use the term gene regulatory code, I only refer to a code that governs these upstream events. Afterwards, there are other points where regulation can interfere: transcription, elongation, termination, RNA splicing, degradation, export, then comes translation, and the activity of the protein may depend on post-translational modifications, multimerization, transport, localization, and again degradation. So this is about essentially regulation of transcriptional initiation. I have to say a few words about current views of eukaryotic gene regulation. Here we have a diagram on top of the slide where a so-called activator protein binds to a DNA element which is called enhancer. Somewhere downstream there is a region called promoter, and then comes a region labeled as a transcribed region. Now, the essence is that we believe that gene regulation doesn't happen at promoters in eukaryotes, unlike a concept that came from bacterial gene regulation. And so it depends on genetic elements that may be far away from the promoter. And if I say far away, it can be several megabases. So it's really long. It's not just a few. It can involve several. It can be a distance which involves many genes. And the most popular model, maybe not the universal model, how this is achieved, is the loop model, which is shown in the center of the slide, where actually a kind of a protein bridge involving the activator protein, a mediator, and RNA polymerase holds the loop together. Now, I do not use exactly the same terminology because it's, for my taste, too specific. So I will, rather than talking about enhancers, I will use terms like cis-regulatory elements, which can also be a silencer. They can have repressive functions. The activator protein for me is a sequence-specific transcription factor. But again, it can have repressive functions as well. The mediator is quite a useful term, I think, but I understand it as any protein that in some way relays information originating from the interaction of a transcription factor with a cis-regulatory element to the actual transcriptional machinery. This is to remind you that there are processes upstream of transcriptional initiation, and those basically relate to switching of chromatin from an inactive state to an active state. And briefly, the active state is accessible to transcription factors, and the inactive state only to maybe a very few transcription factors. So the closed or inactive state is characterized by DNA methylation and by certain histone modification, and the open state is characterized by histone modifications including H3K4 ME3 and H3K4 acetylation. Now, what are sequence-specific transcription factors doing? I say they basically control all the processes that lead to transcription initiation. Also, the switch between open and closed chromatin and histone modifications and the like. Now, they typically consist of two domains, a DNA binding domain and an effector domain. The DNA binding domain recognizes a DNA motif, which is typically about 5 to 20 base pair long, and the effector domain interacts with mediator proteins. They are sometimes called non-DNA binding transcription factors. They also interact with chromatin modifying enzymes, including DNA methylases, components and histone modifying enzymes, components of the transcription machinery, and chromatin structural proteins like histones, for instance. I should say a few words on the techniques that are available to study these processes. And we relatively recently we saw the advent of high throughput technologies which would characterize certain features genome-wide. So chromatin accessibility can be assayed with DNA-seq or ATAC-seq, DNA methylation with bisulfite-seq, histone modification with ChIP-seq, and TF binding, to specific targets also by ChIP-seq and related techniques. Now, I think the important thing to know is that these techniques are genome-wide, so at once they produce a complete map, huge amounts of data. Then we have also techniques that allow to study more downstream effects of gene regulation. So we can assay the tissue specificity of this regulatory element by reporter gene assays. I will show an example. And the in vivo function can be studied on the one hand based on naturally occurring variants, SNPs, but also very recently by gene editing techniques. There are some caveats or important limitation. One is that the chromatin state or TF binding, there are no direct evidence for action or meaning. You know, if a protein binds to a DNA sequence, it may just do nothing. It doesn't mean that it's going to interact with a promoter. And I think with phenotype analysis, the big problem is that we look something that is very far downstream for the actual molecular decoding events. So this is a genome browser view of a genome region. This is just to illustrate the data. We see here in the middle, we see two genes, and they are expression of these genes is driven by divergent promoters. The promoters, so the gene on the left side is transcribed in the opposite direction. So we have two promoters, and between the two promoters we have a bidirectional promoters. That is a typical regulatory, not really a regulatory region, but it's the region that interacts with the regulatory region. On top we see a histone modification track. It's H3K4 ME3. And the peaks you see on the left side, which are spaced by 200 base pairs, they actually represent nucleosomes. And further below we have these horizontal bars, which represent open chromatin regions and transcription factor binding regions identified with ChIP-seq. You see small light green bars in in the gypsy track, and they are actually the motifs that are supposed to bind to the transcription factors. Now these regions are pretty large for one thing, because gypsy has a resolution of about 200 base pairs, but also they actually are merged regions from many assays applied to different cell types. Now, this browser snapshot shows you about 2 KB. So, in terms of the amount of information, the whole genome is more than 1 million times more information. So we really have huge amounts of detailed information, and of course the tracks are only a small fraction of the data that are available. Now I start to develop my analogy between the genetic and the gene regulatory code. So I'm not going to explicitly repeat the elements of the genetic code. So the elementary input unit of the GR code are transcription factor binding sites. There are higher order units, cis regulatory elements. They would correspond to an open reading frame in the genetic code. Now, do we have a parsing machinery like the ribosome, which reads one triplet at a time and then moves to the next triplet? I don't know any. We don't know any. Other translator protein include these mediators. They exist, and they probably would correspond to the amino acid tRNA synthesis in the case of the genetic code. And the global output is not, the elementary output would be a kind of an activating or repressing signal to a gene promoter of transcription factor binding site, and the global output transcription initiation levels of one or several genes. One of the problems with the gene regulatory code is that it's not so easy to just describe the output like in the genetic code. Now, more from a linguistic perspective, so in the genetic code we have words, in the gene we have codons, and in the gene regulatory code the words are the transcription factor binding sites. The open reading frame corresponds to the cis regulatory regions. And positioning rules, it's not clear to me whether we have, maybe we observe some orientation and distance constraints, and it's also not so clear whether we have something like which corresponds to the punctuation signals of the genetic code, which are initiation, termination codons and splice signals. Maybe nucleosomes play a role, maybe not so much. They are somewhat enigmatic insulator elements. Now, a comparison between codons and transcription factor binding sites. Codon is an exact sequence. The TF binding motif is a degenerate motif. The codons interact with tRNAs, and the DNA motifs, they interact with transcription factors. The length is somewhat different, but both the length of the codons are fixed, and transcription factor binding sites 5 to 20 bases. Codons are embedded in an open reading frame. The embedment of transcription factor is not, binding sites is not obvious. Maybe the mechanism is random to access them. There's no combinatorics with the codons. There's combinatorics of transcription factor binding sites through mono and heterodimerization. The codon interact with a single or synonymous adapters, and the transcription factor binding site potentially can interact with non-synonymous transcription factors due to the degeneracy. Now also the number of words is different, you know, genetic code 64, 61, and TF binding motifs, many more than 1,000. Oh, here. Oh. Okay. So this is just a very brief presentation of the degenerate motifs. The standard model is the position weight matrix, which is a table of probabilities to find bases at specific positions, which is often represented by the sequence logo, where the height of the column is actually related to the specificity of the column. The relative heights are still proportional to the occurrence probabilities. Now, it is important to understand that the base probability matrix enables us to compute the measure of binding strength. Such a binding strength can be computed for any sequence, but obviously only the high ranking are relevant for binding. Now, universality and plasticity. So the genetic code is fixed within a species and almost universal among species. The decoding machinery is the same for all cell types, although there may be synonymous components. And overlapping codes are very rare, but they are known and demonstrated. For the gene regulatory code, there is plasticity, there is variability between organisms and cell types. That's speculation again, but that's what we assume. The decoding machinery is variable between cell types. That's the great difference, you know. And overlapping codes are presumably pervasive. Now I want to emphasize the gene regulatory code is cell type specific. It's not the same within a cell. Now this is just an example to remind you of overlapping genetic codes. It's from a phage where you see how the same nucleotide sequence is translated in all three different frames in different proteins. Now, for transcription factor binding sites, the arguments are more theoretical. So this is a slide from last year's Code Bio presentation, where I show that sequence logos, degenerate DNA motif, can be arranged in overlapping fashion in the same DNA sequences. So this is purely, actually behind this is a pure mathematical operation which transforms the lower motifs into the upper sequence. Now, do we know that this exists in nature? Yes, we do. This is an example, again, it's a genome browser snapshot. This is an example which tells us that a short DNA sequence was found in different experiments to bind either to ELF1 or to STAT1 and potentially also to STAT2 and STAT3. And if you below you see the DNA motifs that are recognized by the two transcription factors, and you see that they partly overlap. And if you look carefully at the upper sequence, then you will see that this is kind of the optimal compromise that combines these two motifs. So it exists. Whether they do something, of course, we don't know. Now, reporter gene assays can illustrate another aspect of code variability between tissues. Now, in the reporter gene assays, you use a construct that is shown in the middle of the slide, which consists of a cis-regulatory element that we would like to test, then which is linked to a standard promoter, which then is linked to a reporter gene. Now, a reporter gene is typically an enzyme which transforms a chemical compound into a visible dye. And these constructs are inserted into zygotes, where they get incorporated in the genome, and then the activity of the reporter gene can be followed during development. Now, in this case, 2 cis-regulatory elements have been assayed, and you can see that they drive transcription of the reporter gene at different places of the mouse embryo. So that's what I mean, that this regulatory information is decoded by tissue-specific codes. You see that also these elements are inactive in large part of the embryos. And this again is also an example which shows how unable we are to predict anything, because nobody could ever predict the spatial expression patterns from the sequence. Now, here is my hypothesis of how the gene regulatory code changes during development. So the first assumption is that the code is defined by a subset of transcription factors, a subset of possible adapters. There are over 1,000 transcription factors, and maybe only 100 are active in a given cell type. The same CREs then change meaning after the change of the code by changing from an active into an inactive state, repressive versus activating effect, and also by acting on different target genes. Now, it is important to understand the CREs must themselves participate in the code recoding, in that they are at the origin of activating and repressing new genes, and once these new transcription factors are present, they must work in the proper way. So the code is stabilized, must be stabilized between feedback loops, between CREs and adapters. And typically, the recoding of the code requires some stimulus or the activation of new genes. And we can actually view development as a cascade of temporally and spatially controlled code recoding events. So, do we have some evidence for this hypothesis? I would say maybe the most impressive support comes from a landmark experiments where a research team was able to recode fibroblasts into pluripotent stem cells by activating 4 transcription factors. So that, and after activating these transcription factors, the regulatory state is completely changed, and typical embryonic undifferentiated embryonic transcription factors are activated. Now, I think time is running out, so the last part would be on evolution, and on this slide I wanted to say that the DNA binding specificity doesn't change during evolution. This slide illustrates that the regulatory function may change, and here I wanted to show that some coding sequences are conserved throughout the animal kingdom, whereas regulatory sequences are more strongly conserved between distant vertebrates, but outside it's completely finished, indicating that maybe if they would have to function outside, they would have to obey to a completely different code. This I have repeat again, and here are some thoughts you can about why so little progress has been made on cracking the gene regulatory code. And I think the top and the bottom sentence are the most important one. Research has focused on molecular mechanism rather than meaning, and research hasn't been guided by code biology paradigm.
If you have a question, or okay.
Yeah.
First of all, thank you very much. It was very interesting, and I think we should follow after that. And I have seen immediately some connecting points in these overlapping codes you was talking about. We did last year together with Christian Michel, who is from France too, work about so-called circular mixed sets. Means it is not a code that you can write it in several ways, not only read it in several ways. Maybe we can speak after that about all these topics. Very interesting. Thank you very much.
Sure.
If there are no other questions, I would...
I think there is one.
Oh, sorry.
So, just trying to put it into the wider perspective of code problematics. Can we, just from my understanding, could GR code be seen as a sort of meta language which is applied on, so meta code which is applied to regulator as a regulative system? Could the regulatory code, gene regulatory code, be seen as a meta language in relation to the to the normal genetic code? Is it a meta language, the language which defines its kind of rules of use? Yes, no?
Probably, yeah. I'm not sure whether I know exactly what a meta language would be.
Well, meta language is the language which defines the rules how you use the usual one. For example, well, those are not the rules which are inherent in language itself. For example, there's an American philosopher, H.P. Grice, which created implicatures for the language. So, for example, be brief, be relevant, and so on. So, that's the meta language. How to use the language in communication. Yeah.
Well, maybe what comes to my mind is now that, you know, if I say the code is tissue-specific and it is defined by a subset of adapters, transcription factors. This subset can be split into two parts. The one is which defines the regulatory state itself. So that's factors that somehow are involved in feedback loop. And then there is the other part which define the target genes of the state. And so I would say the first one is the meta language, and the second one is just the lower level language. Yeah.
Just to follow up with my colleague here about the regulatory code. There is clearly a variation, as you said, a variation between species. Is there? I mean, it seems that there is a fixed part and there is a variational part. No, and this is exactly coming from linguistics. We have this universal part and parameter, at the parameter level. But we do have a relation between these two things. So my question to you is, at the regulatory code, the variation itself, is there a pattern you can predict between species, or there is nothing, no rules governing this variation at the regulatory code?
Well, you are speaking about differences between species.
Yes.
I do not think there is a fixed part. I think it's completely different between species. The code as such. You know, the molecular components, they are not. But yeah, it's difficult to identify globally. I mean, maybe it's, well maybe by reference to the genetic code, a few codons are shared, yeah. But yeah, you could see it like this.
Okay, thank you.
Yeah.